In this video, we will discuss the DNA replication process. It occurs in the S phase of the cell cycle. Okay, so we will come to the step-by-step process. Consider one DNA fragment. This has to be broken down. Now it is broken down and we can name it as A strand and B strand. These are the parent strands. Now on this A strand and B strand, a new strand has to be synthesized, one here and the other one here. So I'm labeling it as A dash and B dash. So A dash and B dash are the new strands, which is red in color, while A and B are the parent strands. So now from one DNA fragment, two DNA fragments are formed. It doesn't occur as like this. It is not all of a sudden process. It is a step-by-step process. So we will discuss each and every step. In the beginning, these nucleotides has to be there in the cell, and they have to be activated. So that will be the first step, the activation of these nucleotides. Each one will be a nucleotide, okay, because this is a polynucleotide chain. So these nucleotides have to be activated because energy is required for binding process. So that will be the first step. So it is an activation of deoxyribonucleotides. And this deoxyribonucleotides can be also called as adenine monophosphate, guanine monophosphate, etc. So these are the deoxyribonucleotides. These are the other name. Now they have to be activated. So they will be converted to their triphosphates. So now these triphosphates are made from AMP to ATP, GMP to GTP, like that. Okay, they will be present in the cell. Now this has two purposes. One, they act as the substrate for making new strands. Second one, they provide the energy for this polymerization reaction. Okay, so this is the first step. Next we can go to the second step. So here one DNA fragment is there. The strands has to be broken down one by one from one side. So suppose if it starts from this side, we can call it as this side as origin of replication, or in short it is ORI. So this is origin of replication or ORI, this region. Okay. Now here that region has to be recognized. It is recognized by a protein which starts this process. So we call that protein as initiator protein. Okay, this initiator protein will come and make a nick here, that is a break here. Okay, so that region will be recognized as ORI. Now from this point, the replication starts. Okay, so the second step is the recognition of this initiation point. It is done by initiator protein and that region is ORI. So this is the second step, recognition of initiation point. Now this hydrogen bonds has to be broken down. So one protein will come and bind here, and it will go on breaking the hydrogen bond. That is called unwinding protein. So that will be the next step, third step. So here the unwinding protein come and bind here, then it will go on breaking these hydrogen bonds. So after some time these are broken. Now it has reached here. Now it will go in this direction. So as it proceeds, it reached here. Okay, so it has broken this much bonds. Now this area look like a fork, and it formed during this replication process. So this is called a replication fork. So this is the third step, unwinding of DNA. It is done by unwinding protein, for example helicase. Okay, so a replication fork is also formed. Now here new DNA strands has to be synthesized. Okay, so for knowing I'm writing the ends here. If this is 5' and this is 3', then this will be 3' and this will be 5' because of anti-parallel polarity. Same here also. Okay, now the new strand is synthesized in 5' to 3' direction. Okay, new strand is synthesized in the direction 5' to 3' direction. That is in this strand. Okay, this is 3' end of the parent strand. So new strand will have here 5' end. So in this direction it will be synthesized. But here this is 3' and this is 5'. So you consider only this strand. So if this end is 5', this end will be 3'. Okay, so this will be 5'. So here the direction of synthesis will be this. It is done by DNA polymerase enzyme, but it cannot start the synthesis. It needs some help. So the help is done by an RNA strand, which is called Rna Primer. So Rna Primer will come and attach to the starting point. Here it is in this direction because this is 3'. So new strand will have this as 5'. Okay, so this will be the starting point. Here this will be the starting point. So this RNA strand has RNA primer, that is small RNA strand, has to be binded here. It is done with the help of an enzyme, RNA polymerase, but it is directed by the DNA. So we can call it as DNA directed RNA polymerase enzyme. That enzyme helps in binding RNA primer, because DNA polymerase cannot initiate the process. So the RNA primer will help it. So that is the next step. So this is the next step, attachment of RNA primer to the 3' of old strand or parent strand or 5' end of the new strand. Okay, and it is done by DNA directed RNA polymerase enzyme. Pol is the short form. Okay, next the new strands can be synthesized here. So this is the next step, synthesis of new DNA strands. And always it is in the direction 5' to 3'. So here in this direction it will be made, this direction, while here in this direction it will be made. Now as this process continues, the unwinding protein will go on unwinding in this direction. Okay, now here this is the direction, because these are the ends. This is 5', 3', here 5' to 3', it will go on synthesizing. So whenever it get a new bond, it will go on synthesizing very easily. Okay, but here the direction is this. Okay, because this is the 5' end, this is the 3' end. So it will wait for some time for getting some more free hydrogen bonds. But here it will not wait, it will continue, because the unwinding is in this direction, the synthesis is in this direction. So it can easily continue whenever it get the hydrogen bond. But this side it will wait for some time. And here since it is in this direction, again it has to start from here. So here a new RNA primer will bind. Okay, then only it will make the new DNA strand. So here a new DNA strand is made. Okay, so in this side it is a continuous strand, while in this side it is a discontinuous strand. So this strand is called a continuous strand, this is called a discontinuous strand. So this is continuous. Okay, and this will lead this process, because whenever it get a new hydrogen bond it will make. So it is also called leading strand, while here this will stop for some time, because it has to get few more hydrogen bonds. Okay, so it will be lagging. So this is discontinuous or lagging strand, this is continuous or leading strand. So here in this one you will get small fragments here. These fragments of DNA are there, because it is discontinuous, and these fragments are called Okazaki fragments. Okay, so in this synthesis of new DNA strand you have to write about continuous strand, discontinuous strand and Okazaki fragments. And these new DNA strands are being synthesized by the nucleotides has to be attached. It is attached by DNA polymerase. RNA was attached by RNA polymerase enzyme. So DNA strand will be attached by DNA polymerase enzyme, but it is dependent on this DNA, parent DNA. So we call it as DNA dependent DNA polymerase. So this is the name of the enzyme, DNA dependent DNA polymerase enzyme. So you have to write these much things in synthesis of new DNA strand. Now we will continue to the next step. So as this process continues, we will see what will happen. You can see when the whole unwinding is over, in this side the new strand will be continuous strand 5' to 3', but here you will get many Okazaki fragments. Now in the next step, this RNA primer function is over, it has to be removed. So it is removal of RNA primer. It is done by DNA polymerase enzyme itself. Okay, so it is done by the exonuclease activity of the DNA polymerase enzyme. Okay, so DNA polymerase enzyme will come and cut the RNA primer and remove it. So we call it as exonuclease. This strand has been removed. Okay, now when after removing you will get many Okazaki fragments, it has to be joined. That is the next step. So one strand is okay, but in this strand you will get many Okazaki fragments. This has to be joined. Okay, it is joined by an enzyme, DNA ligase, because the ligation is the name of the joining process. So it is joined by DNA ligase. Okay, so DNA ligase will come and join these DNA fragments. Okay, now as the last step, the DNA polymerase will come and check this whole DNA fragment, newly synthesized one, whether any nucleotide has been misplaced. Okay, so here it will come and read the proof, that is proofreading, and if any problem is there, if any misplacing is there, then it will do the DNA repair. This will be done by DNA polymerase enzyme, but for joining it requires DNA ligase enzyme. So in the last step, two enzymes are there. DNA polymerase and DNA ligase enzyme. Okay, so this is the DNA replication process. Thank you.